Right now, in a pitch-black incubator kept at exactly 37 degrees Celsius, a cluster of, roughly 100 microscopic cells is about to be evaluated.
Mm-hmm. And the score this tiny biological structure gets on the next 60 seconds, it's going to dictate its entire future.
Yeah, literally its entire future. That score determines if it's selected to become a human life today or if it gets submerged in liquid nitrogen.
Yeah,
Just, frozen in suspended animation for a decade.
exactly. And welcome to this deep dive, by the way. Today, we're really immersing ourselves in the fiercely guarded, high-stakes environment of episode 12 of the Inside the IVF Lab series.
Right, the one titled Choosing the Embryo, Selection for Transfer. And the gravity of that 60-second evaluation we just mentioned, I mean, it really cannot be overstated.
Oh, absolutely not. We're looking at a process that requires just this immense confluence of patience, precision, and honestly, an almost forensic level of biological analysis.
Yeah, forensic is a good word for it. We're shifting the narrative away from that sort of romanticized idea of passive observation, right, where embryologists just kind of watch cells divide.
Right, because the reality is highly rigorous. It's an active, heavily data-driven prioritization process. It's not just sitting back and watching.
So to navigate all this, we are unpacking a really phenomenal stack of source materials today. We've got detailed laboratory notes, comprehensive training guides.
Well, ones used to calibrate the eyes of new embryologists, yeah. Those are fascinating.
They really are. And we also have the uncompromising clinical protocols
Sure.
that dictate decision-making in top-tier fertility clinics. So if you were listening to this, you might be an embryology trainee yourself.
Yeah, maybe spending hours in a dark room learning to identify all these tiny cellular anomalies.
Or you might be a clinician who actually relies on this exact data to counsel your patients. Or, you might be an IVF patient who is intimately familiar with that agonizing wait for the day 5 lab update.
Ugh, that wait is brutal. But regardless of your background, our mission today is to decode the exact cellular mechanisms and structural criteria that come into play when the perfect choice just isn't obvious.
Okay, let's unpack this.
Mm-hmm.
We really need to start by setting the biological clock, I think. We're dropping right into the critical juncture in the incubator, which is day 5 or day 6 of embryonic development.
Yeah, and to fully grasp the pressure of day 5, we kind of have to look at the metabolic and physical transformations that lead up to it.
Like the gametes in the very beginning.
Exactly. In the beginning, the embryologist is managing individual gametes. They evaluate the maturity of the egg, the oocyte.
Right.
They're tracking its progress, making sure it has extruded its first polar body. That signals its transition from an immature M1 state to a fully mature, fertilizable M2O oocyte.
And once fertilization is actually confirmed, for the next, 120 hours or so,
Mm-hmm.
the embryologist's primary operational mode is really just being a caregiver.
Very much so. It's a highly protective environmental control protocol.
I mean, they're managing the biochemistry of the culture media, right? Ensuring the precise shift from the pyruvate-heavy solutions that support early cleavage.
Mm-hmm. To the glucose-dependent media that's required for later blastocyst development. It's this intense exercise in strict environmental homeostasis.
They're basically acting as a surrogate fallopian tube in uterus.
Literally, they are shielding the cells from any fluctuations in pH, temperature, even volatile organic compounds in the air. But then, day 5 arrives.
Then a profound biological threshold is crossed. The embryo undergoes compaction and cavitation, right?
Yes. It transforms from this loose cluster of cleavage stage cells into a highly organized, differentiated structure called a blastocyst. And at this precise morphological milestone, everything changes.
The embryologist's role undergoes a severe pivot. They have to instantly drop the caregiver persona and, become a judge.
A very critical judge. And that transition from nurturing every single embryo to actively scrutinizing and ranking them, it requires a massive psychological recalibration.
I can imagine. For 5 days, the absolute goal has been collective survival for all of them.
Exactly. And now they're peering through the optics of an inverted microscope at maybe 5 or 10 developing blastocysts, and they have to run a ruthless biological triage.
To answer just one question, right? Which single structure possesses the highest metabolic and structural competence to establish a pregnancy?
That's the one. And this brings us to a fundamental biological truth that governs the entire laboratory framework. It's the concept of selection versus creation.
Selection versus creation. I mean, the IVF laboratory, even with the most advanced time-lapse incubators and specialized culture media, it possesses zero ability to actually improve the inherent genetic architecture of an embryo.
Right, they cannot create a perfect embryo from flawed constituent parts. It's
Right.
just not biologically possible.
So if the DNA contains a fatal structural error, or, if there's a heavy fragmentation burden inherited from that M2 oocyte, the lab is entirely powerless to reverse that.
Completely powerless. They aren't manufacturing a product on an assembly line here. They are fundamentally constrained by the biological raw material provided by the patient.
Which means the most powerful tool the lab wields at the end of a cycle isn't enhancement at all. It's purely selection.
Exactly. Their mandate is to accurately identify the highest potential within the existing cohort. And the pressure of this shift from caregiver to judge, it's really mitigated by relying entirely on objective data.
Quantifiable data matrices. Because if an embryologist allows, say, emotional attachment to influence their grading, that would be a disaster.
Oh, completely. Like if they favored an embryo just because it struggled early on but eventually formed a blastocyst, the clinical outcomes would plummet.
Right. It sounds like transitioning from being a gardener, nurturing a greenhouse full of plants, to suddenly being a food critic, having to choose the single best apple from the harvest. Is that sudden shift jarring for the scientists?
Well, what's fascinating here is that the pressure of that pivot is managed through strict objective data rather than emotion. They have to be objective.
The source materials actually explicitly refer to this selection process as an efficiency tool, which honestly is a fascinatingly clinical way to describe something so deeply personal to the patient.
It is clinical, but in reproductive medicine, efficiency translates directly into minimizing a patient's exposure to failed transfers.
And the physiological and psychological toll that comes with those
Well,
failures.
exactly. It is entirely about shortening the timeline to a successful live birth. The clinical strategy is always centered on minimizing the number of interventions.
So if a patient generates 5 viable embryos, several of those might actually possess the intrinsic competence to yield a live birth, right?
They might, yeah. But the embryologist's task is to analyze the microscopic data to predict which one will implant the fastest. And we also have to distinguish between the urgency of a fresh transfer versus cryopreservation.
Right, because with a fresh transfer, you're racing against the patient's collapsing window of implantation.
Yeah, the endometrium is only receptive for a highly specific number of hours.
Driven by the patient's progesterone exposure, right? The embryo's developmental stage has to perfectly synchronize with that uterine environment.
Whereas with cryopreservation, the logistical pressure is completely different.
How so?
Well, the lab evaluates the cohort to determine which embryos have the structural integrity to survive the severe osmotic stress of vitrification.
The ultra-rapid freezing process.
Exactly. They are effectively hitting the pause button on biological time. That allows the clinician to prepare the optimal uterine environment weeks or even months later.
Wow. Okay, so we understand the immense pressure of the pivot. The embryologist is now the judge. But a judge requires a codified rubric to evaluate the biological contestants,
They absolutely do.
which leads us into the architecture of the grading system. Because how do we quantify the chaotic beauty of cellular division into actionable data?
Well, the global standard is the Gardner grading system. It was designed to standardize visual morphology.
To transform subjective cellular observations into a universal alphanumeric language. So an embryologist in Tokyo can understand the exact developmental stage of an embryo graded by an embryologist in New York.
Exactly. It's a universal language. And the system utilizes a 3-part code, which is a number followed by 2 letters.
Okay, let's dissect this code systematically because this is the exact data patients spend hours agonizing over on their lab portal. I know I would be hitting refresh constantly.
Sure. So the first variable is the number, which ranges from 1 to 6. This quantifies the degree of blastical expansion and the hatching status.
Okay, to visualize this, we have to understand what drives expansion in the first place. As the embryo transitions into a blastocyst, the outer layer of cells begins actively pumping sodium ions into the center of the embryo, right?
Right. And through osmosis, water follows that sodium. It creates a fluid-filled cavity called the blasticle.
So the expansion number is essentially a measurement of how efficiently those microscopic cellular pumps are working.
Yep. So a grade of 1 or 2 indicates very early cavitation. The pumps have just turned on, and the fluid cavity is less than half the volume of the embryo.
Got it. And as those sodium-potassium ATPase pumps continue to work, the cavity swells.
Exactly. A grade 3 is a full blastocyst, where the fluid cavity completely fills the embryo, but grades 4, 5, and 6, those represent critical mechanical milestones.
Okay, let's go through those. What's a 4?
A grade 4 is an expanded blastocyst. The fluid pressure has built up so intensely that the outer protective shell, the zona pellucida, is being physically stretched and thinned out.
So the embryo is literally outgrowing its biological container.
It has to if it wants to implant.
Hmm.
And then a grade 5 indicates that the embryo has actually initiated the hatching process.
It's breached the zona pellucida.
Yes.
Yeah.
Usually through a combination of physical pressure and the secretion of proteolytic enzymes that dissolve a small hole in the shell. The embryo is actively herniating out.
Wow, just squeezing its way out. And a 6.
A grade 6 means the hatching process is completely finished. The blastocyst is entirely free of the zona, and it's ready to make direct physical contact with the uterine lining.
Okay, so the numeric value gives us the fluid dynamics and the hatching status. That makes sense. But the letters dive into the specific cellular populations. The first letter in the sequence, A, B, or C, evaluates the inner cell mass, the ICM.
Right. The ICM is this densely packed, distinct cluster of pluripotent cells attached to the inside of the blastocoel cavity.
And these are the cells that possess the remarkable capacity to differentiate into every single tissue type in the human body. The ICM will ultimately become the epiblast and then, the actual fetus.
Exactly. So when an embryologist assigns an A to the ICM, they're seeing a highly cohesive, tightly packed aggregate containing a large number of distinct cells.
It represents a really robust, morphologically pristine foundation for fetal development then.
Precisely. While a B grade indicates a slightly looser configuration, maybe fewer cells overall, but it's still highly viable.
And a C's grade.
A C grade signals a noticeable deficiency. It might be a very sparse cellular cluster, or cells that appear granular or necrotic, which statistically correlates with a lower probability of sustained fetal development.
Okay. And the final character in the code, the second letter, that evaluates the trophectoderm, the TE. So if the ICM is this clump inside, the trophectoderm is the spherical, single-cell, thick epithelium forming the outer wall of the blastocyst.
Yes, and these are the cells responsible for the heavy lifting of early pregnancy. The trophectoderm is tasked with adhering to the endometrial lining, invading the maternal stroma, and eventually developing into the placenta.
It's basically the embryo's life support system and its communication array. It secretes HCG to signal the maternal body that a pregnancy has even begun.
Right. So again, an A grade here signifies a highly organized, contiguous epithelium composed of many small, healthy cells forming a really strong barrier.
And a B indicates fewer cells or a slightly irregular cellular matrix, while a C suggests a fragile, very sparse outer layer. I'm trying to visualize the mechanical relationship between these three variables. It kind of feels a bit like inflating a hot air balloon.
Oh, I like that. How so?
Well, the expansion number is the volume of hot air being pumped in, dictating the overall size and tension. And the fabric itself, the physical barrier holding the structure together, is the trophectoderm, the second letter.
That is a highly accurate physical model. The hot air stretches the fabric of the balloon.
Right. And then the payload, the tightly packed basket hanging securely inside, is the inner cell mass, that first letter. You need the air pressure to expand the structure, a strong fabric to maintain integrity without tearing, and a secure basket to carry the payload.
That's exactly it. And if the fabric, the trophectoderm, is heavily degraded, resembling a C's grade, the balloon cannot maintain pressure, regardless of how pristine the payload inside might be.
Which raises a critical clinical debate, actually. If I'm looking at a 4A versus a 4AB, am I prioritizing the fetus cells, the ICM, over the placenta cells, the TE? Does the knot matter more than the rubber?
It's a great question. In the early days of IVF, the assumption was always that the fetus was the absolute priority. Intuitively, it makes sense to focus on the ICM.
Right, because that's what becomes the baby.
Exactly. But modern transcriptomic and implantation data have radically shifted our understanding here. The biological reality is that implantation is an aggressively invasive event initiated entirely by the trophectoderm.
So if the TE lacks the cellular density or metabolic energy to breach the uterine epithelium and establish a vascular connection, the embryo just arrests. The pristine ICM will never have the opportunity to develop.
Exactly. The payload doesn't matter if the balloon crashes before it can even land. A robust trophectoderm is an absolute prerequisite for survival.
Wow.
In fact, some contemporary clinical data even suggests that trophectoderm grading might be a stronger independent predictor of live birth rates than the ICM.
Really? Just because early implantation failure is the most common bottleneck in IVF.
Yep, exactly. It's the biggest hurdle.
To actually see these microscopic differences, we have to look at the hardware too. The embryologist isn't just holding a petri dish up to a light bulb, right? The lab's Spotlight notes detail the use of highly specialized inverted microscopes.
Oh, the tech is incredible. They utilize inverted microscopes equipped with Hoffman modulation contrast, or differential interference contrast, DIC optics.
And what does that do exactly?
It manipulates the light path to create a pseudo three-dimensional image of these transparent living cells without requiring toxic chemical stains.
That's wild. So the light source sits above the sample, and the objective lenses are positioned below, observing the embryos through the optically clear bottom of the culture dish.
And these optical feeds are piped directly into high-definition monitors. The days of squinting through binocular eyepieces for hours on end are mostly over.
But even with stunning 4K resolution, grading still relies on human interpretation. How does the laboratory director ensure that an embryologist doesn't suffer from grading drift? Like, how do you prevent one technician's A from becoming another technician's B?
Inter-observer variance is the enemy of a standardized lab. So to combat this, rigorous quality control protocols are implemented.
Like what?
Laboratories utilize extensive digital libraries of reference images, like a visual taxonomy of established grades, and they conduct blind proficiency testing.
So the entire embryology team independently grades a historical cohort of embryos, and then they compare the results.
Exactly. The results are statistically analyzed to identify any deviation. If an embryologist is consistently grading trophectoderms too harshly compared to the laboratory mean, they undergo targeted recalibration training.
It's a constant, ongoing alignment of human perception against a fixed clinical standard. That's intense.
It has to be. So we have this fiercely calibrated team of judges, equipped with advanced optics, identifying embryos with high-grade expansion, robust ICMs, and highly cohesive trophectoderms.
But here is the friction point in the data. If the Gardner system is so highly predictive, and a patient has a cohort containing 3 flawless 4AAs, the immediate instinct, particularly for a patient who has endured immense financial and physical trauma, is to demand that all 3 be transferred simultaneously to mathematically guarantee a pregnancy.
It is the most pervasive and understandable instinct we encounter in clinical practice. Of course, you'd want to maximize your chances right then and there.
Right, but honoring that request essentially weaponizes the precision of our own technology.
It really does. To understand why, we have to examine the historical context of embryo transfer strategies. Back in the 1980s and 90s, culture media formulations were just metabolically inadequate. They couldn't sustain an embryo in vitro past day 3.
And at day 3, you are transferring a cleavage stage embryo. It's just 6 or 8 cells. It hasn't even begun to differentiate into ICM or trophectoderm.
Right. And at that stage, visual morphology is an incredibly weak predictor of ongoing developmental competence. Many butyrially symmetrical 8-cell embryos will just arrest the moment they are required to activate their own embryonic genome.
So because the predictive power was so low back then, the standard practice was a scattergun approach. Transfer 3, 4, or even 5 cleavage stage embryos, relying on the sheer volume of biology to overcome the high attrition rate, just hoping one would survive to implant.
Exactly. But the advent of sequential culture media and single-step continuous media allowed the lab to confidently culture embryos out to the blastocyst stage.
The lab environment effectively became the primary filter. It allowed embryos burdened with lethal genetic errors to arrest in the dish rather than in the uterus.
Yes, and that biological filtering radically increased the implantation rate per transferred embryo. We move from an era of guesswork to an era of high precision.
Consequently, the philosophy of transfer had to undergo a seismic shift. Transferring multiple highly rated blastocysts today doesn't just increase the chance of a pregnancy, it dramatically inflates the probability of high-order multiple gestations.
Which is why single embryo transfer, or SET, became the unequivocal gold standard of modern reproductive medicine.
Because the human uterus is anatomically and evolutionarily designed to carry a singleton. You put 3 4AAs in, you don't magically get 3 safe, isolated pregnancies. You create a dangerous, high-stakes competition for vascular resources and uterine space.
The maternal risks are severe and they cascade quickly. Gestational parents carrying multiples face exponentially higher rates of profound preeclampsia, gestational diabetes, placental abruption, and even catastrophic postpartum hemorrhage.
And the neonatal risks are equally devastating. The goal of IVS is not just a positive pregnancy test. The goal is a healthy, singleton baby born at full term.
I'm so glad you said that, because multiple pregnancies inevitably truncate the gestational timeline. Prematurity is virtually guaranteed with triplets, and it's highly common with twins.
And premature neonates are thrust into the NICU, battling respiratory distress syndrome due to underdeveloped lungs, facing a higher incidence of intraventricular hemorrhage in the brain.
And grappling with long-term neurodevelopmental deficits. The fundamental objective of the IVF laboratory is solely the delivery of a healthy, full-term singleton neonate.
Which makes logical, clinical sense. But if I'm a patient paying for this process, putting all my chips on one number on the roulette wheel feels terrifying. How do you convince someone that holding back their second-best embryo is actually in their best interest?
That is where we have to reframe the timeline and introduce the concept of cumulative probability. The mathematics strongly support the ESET protocol.
Okay, let's map out the scenario. Say you transfer two highly rated blastocysts simultaneously.
Right. Yes, you might achieve a 65% chance of a clinical pregnancy on that single attempt, but you carry a massive 20 to 30% risk that the pregnancy will be twins.
So you're taking on a massive risk liability for a marginal 5% increase in initial success.
Exactly. Now, execute the SET protocol. You transfer the single highest graded blastocyst today. That yields your 60% chance of a singleton pregnancy. The second high-quality blastocyst is vitrified and cryopreserved.
Okay.
If the initial fresh transfer fails to implant, the patient returns for a frozen embryo transfer, utilizing that second blastocyst, which carries its own independent 60% probability.
This is basic binomial probability. You're decoupling the events. By running the events sequentially rather than simultaneously, the cumulative chance of achieving a live birth across those two independent transfers actually climbs significantly higher than the simultaneous double transfer.
It often exceeds 80%. You achieve a vastly superior overall success rate while completely eradicating the neonatal danger associated with a twin gestation. You're stretching their biological potential over a safer timeline.
Maximizing the efficiency of the entire cohort.
Yeah.
Okay, so SET is the firewall protecting the patient from the precision of the lab. But by committing so absolutely to single embryo transfer, the margin for error in selection drops to zero.
It really does.
You are placing the entirety of a cycle's physical and financial weight on a single microscopic structure. And as detailed as the Gardner system is, visual morphology is still just looking at the architectural exterior of the embryo. It tells you nothing about the molecular blueprint inside.
Which is why, to achieve the required level of certainty, the lab has to deploy a selection stack. Visual morphology is no longer the terminus of our diagnostic capability.
It's merely the baseline layer of the stack. To definitively answer the question of which embryo goes first, laboratories integrate profound technological advancements, primarily PGTA, pre-implantation genetic testing for aneuploidies.
Right. And let's break down the mechanics of PGTA because it represents an incredible feat of micromanipulation. The embryologist actually has to perform surgery on the blastocyst.
Wait, they do surgery on it?
Yeah.
The biopsy procedure must be a masterclass in precision.
It is. On day 5 or 6, as the blastocyst begins to hatch, the embryologist uses a highly targeted infrared laser to pulse the zona pellucida, creating a microscopic breach. And then they apply gentle suction using a finely calibrated glass micropipette to aspirate a tiny cluster of cells, usually between 3 and 5 cells, specifically from the trophectoderm.
And they intentionally avoid the inner cell mass to ensure the future fetus remains entirely undisturbed, right?
Correct. The trophectoderm is highly robust and recovers rapidly from the biopsy. Those aspirated cells are sheared off using the laser, loaded into a microcentrifuge tube, and sent to a specialized genetics laboratory,
Where the DNA undergoes whole genome amplification and next-generation sequencing to determine the exact chromosomal complement of the embryo. They're looking for aneuploidy, an abnormal number of chromosomes.
Exactly. The standard human blueprint requires 23 pairs for a total of 46 chromosomes, and PGTA fundamentally disrupted the historical reliance on visual morphology because it revealed a stark biological reality.
Which is that cellular beauty does not guarantee chromosomal integrity.
Yes. An embryo can execute flawless cleavage, form a spectacular blastocoel cavity, and possess a densely packed A-grade ICM, presenting as a textbook for AA. But the sequencing data might reveal a missing copy of chromosome 16 or an extra copy of chromosome 21.
It's an aneuploid beauty
It's
queen.
an aneuploid beauty queen, exactly. And the biology is unforgiving here. An embryo missing a crucial autosome will trigger massive cell cycle arrest or widespread apoptosis. It simply will not be permitted to implant, or it will result in a rapid, early clinical miscarriage.
Conversely, you might evaluate a morphologically modest blastocyst. It's fully expanded, but the ICM is a bit sparse, and the trophectoderm cells are slightly irregular. A standard 3BC or 4BC, visually it's an underdog.
But the genetic analysis confirms it is entirely haploid. It possesses a perfect 46 chromosome karyotype.
In that scenario, the genetic data obliterates the visual hierarchy. The euploid ugly embryo possesses the fundamental biological competence to yield a live birth, whereas the aneuploid perfect embryo possesses zero potential.
PGTA dictates that chromosomal status is the absolute ultimate arbiter of viability.
Here's where it gets really interesting, though. If a genetically normal ugly embryo beats a genetically abnormal beauty queen embryo every time, why do we even bother with the visual Gardner grading anymore?
It's a fair question.
Why not just biopsy every single blastocyst, discard the aneuploids, and throw a dart blindly at the remaining diploids? If PGTA is the background check verifying the applicant isn't a criminal, Gardner grading is the actual job interview to see who is the most charismatic, right? But if the eploid embryo always wins, regardless of how it looks.
Well, if we connect this to the bigger picture, having a clean background check doesn't necessarily mean you're the most efficient worker for the job.
Okay.
The reality is that many patients, particularly younger cohorts or those utilizing egg donors, will produce multiple euploid embryos within a single cycle. You might receive a report indicating you have 4 chromosomally perfect candidates.
So PGTA confirms they all possess the capability to become a child. But it cannot differentiate their metabolic efficiency. It can't tell you who to hire first.
Precisely. It is the morphology that acts as the vital tiebreaker. When genetic competence is equalized across a cohort, the embryo that has established the most robust trophectoderm and the densest ICM is statistically proven to implant faster and with higher efficiency.
We rely on the genetics to establish safety and the morphology to optimize the speed to success.
Exactly. And to refine that speed even further, the most advanced labs employ the final layer of the selection stack: time-lapse morphokinetics.
This technology feels like a massive leap forward. Traditionally, grading required the embryologist to physically remove the culture dish from the incubator, place it on the heated stage of the microscope, quickly execute the morphological assessment, and rush it back to minimize environmental stress.
It was a single, highly pressurized snapshot taken every 24 hours. You capture the state of the embryo at 8 a.m. on day 3, and again at 8 a.m. on day 5.
But you are completely blind to the biological events unfolding during the intervening 48 hours.
Which is where time-lapse incubators come in. They eradicate that blind spot. The embryos remain continuously undisturbed in their highly controlled microenvironment.
And instead, an integrated optic system captures high-resolution digital images of every embryo at 10 to 15-minute intervals throughout the entire 5-day culture period.
It stitches thousands of snapshots together to generate a continuous, dynamic film of embryonic development. You aren't just looking at the final destination, you're analyzing the precise journey.
We call this data morphokinetics. Time-lapse allows us to extract precise chronological timestamps for every significant cellular event.
Right. We can record the exact minute the first cleavage division occurs, usually between 25 and 27 hours post-insemination. We track the duration of specific cell cycles, the time it takes to progress from 2 cells to 3 and 3 cells to 4.
Which completely changes the paradigm, because an embryo that hits the 8-cell stage at 60 hours is biologically a very different entity than one that hits it at 90 hours, even if they look identical under a standard microscope by day 5.
Exactly.
If I'm understanding the biological implications, you're tracking metabolic efficiency.
You hit on the exact mechanism. We are looking for highly synchronous, rhythmic cellular divisions. If the cell cycle length is abnormally truncated, it implies a frantic, chaotic division.
And the time-lapse feed often reveals phenomena that a daily snapshot would entirely miss.
Oh, absolutely. For instance, an anomaly known as direct cleavage. A normal blastomere should undergo mitosis to form 2 daughter cells, but occasionally a single cell will violently fracture into 3 or 4 cells simultaneously.
Just a chaotic explosion of genetic material. That sounds bad.
is, which almost guarantees uneven chromosomal segregation. We also observe reverse cleavage, where an embryo divides from 2 cells to 3 and then aberrantly fuses back into 2 cells hours later.
So if you only looked at the snapshot the next morning, you might see a perfectly symmetrical, normal-looking 4-cell embryo, completely unaware that it arrived at that state through a catastrophic abnormal pathway.
It effectively hides its chaotic history. And embryos that exhibit direct cleavage or significant reverse cleavage demonstrate severely compromised implantation potential, even if they manage to eventually form a decent-looking blastocyst by day 5.
So the time-lapse kinetic data allows the embryologist to deprioritize or completely discard embryos that mask underlying metabolic or structural turbulence, ensuring the embryo chosen for set is both genetically sound and metabolically rhythmic.
Exactly. So we have the ultimate master plan. We have the foundational Garger morphology. We have the PGTA chromosomal background check. And we have the time-lapse kinetic film confirming a flawless developmental journey. The spreadsheet is complete. The top-ranked embryo is locked in.
The clinical strategy is fully formulated, but, the biological reality of transfer day introduces an element of controlled chaos. Living tissue does not adhere to administrative spreadsheets.
It definitely doesn't.
The source notes detail how dynamic and volatile the actual morning of the transfer can be. It is an environment defined by rapid, real-time recalculation. The absolute truth of a blastocyst is that it is a highly active, fluid-pumping entity.
Right. And the morphological state of an embryo at 8 a.m. can differ radically from its state at 11:30 a.m. when the patient is prepped for the procedure.
Walk me through the chronology of that morning. The patient's transfer is scheduled for 11 a.m. What is the embryologist doing at 7:30 or 8 a.m.?
The morning begins with a comprehensive reevaluation of the patient's specific cohort. If they are utilizing frozen embryos, the selected blastocyst is carefully thawed and its survival and re-expansion are monitored.
And if it's a fresh transfer cycle?
The embryologist reviews the time-lapse feeds, collates the genetic reports if rapid PGTA was performed, and executes a real-time morphological grade. They synthesize this data matrix to generate the final rank list.
They confirm the primary target and establish a backup contingency.
Once the hierarchy is established, the critical communication loop is initiated. The embryologist conducts a pre-transfer briefing with the reproductive endocrinologist, the physician executing the medical procedure.
This ensures absolute alignment between the laboratory's microscopic data and the physician's clinical approach before the patient even enters the facility. They confirm the embryo's grade, its kinetic history, and verify the patient's identity protocols.
But as the clock ticks toward 11 a.m., the patient is now in the procedure room, the speculum is placed, and the physician calls for the embryo. The embryologist returns to the incubator to retrieve the primary target to load it into the transfer catheter.
And this is the moment where the dynamic reality of biology asserts itself.
This is where the tension spikes. You retrieve the Petri dish, place it under the optics, and observe that the primary target, an embryo that was a brilliantly expanded 4AA at 8 a.m., has suddenly, spontaneously collapsed.
Collapsed? I mean, if I have a patient lying on the table and I hear the word collapsed whispered in the lab, my immediate physiological response is sheer panic. It sounds like a catastrophic structural failure.
It is an alarming term for a layperson, but to an embryologist, a collapse is frequently a routine physiological event. It is deeply tied to the mechanics of hatching.
Okay, how so?
Well, the blastocyst is constrained by the rigid zona pellucida. To herniate through the small aperture it has created, the embryo must alter its physical dimensions. The trophectoderm cells intentionally disrupt their tight gap junctions, allowing the accumulated fluid in the blastocoel cavity to rapidly leak out.
It sounds like a live sports draft right up until the buzzer. How nerve-wracking is it for the embryologist to change the game plan at 10:55 a.m. because an embryo suddenly decided to collapse or hatch? It purposely expels its internal volume to shrink its physical footprint. It deflates itself to squeeze through the exit door.
It's a calculated biological maneuver. However, if the embryologist observes the embryo at the exact nadir of its collapse, it appears as a tight, disorganized cluster of cells. The beautiful blastocoel is gone. The cellular architecture is indistinguishable. It no longer resembles a 4AA.
So the embryologist is staring down the microscope, holding the transfer catheter, and they have to make an immediate high-stakes judgment call. Do they trust the historical data, the 8 a.m. grade, and the kinetic film, and load the collapsed embryo, knowing it's a normal physiological phase? Or do they pivot to the number 2 embryo on the rank list, which is currently fully expanded and visually pristine?
It demands an exceptional level of clinical composure. They must rapidly synthesize the immediate visual data against the historical kinetics. If the time-lapse feed confirms the collapse initiated just 30 minutes ago as part of a hatching pulse, they will confidently proceed with the primary embryo.
But if the collapse appears atypical or if the cellular debris looks necrotic, they will unhesitatingly switch to the backup.
Exactly. And then there is the intense scenario of navigating absolute ties.
Right. What happens when the variables are completely deadlocked? Say a patient has 3 euploid embryos. Their kinetic timelines are virtually identical, and they are all graded as 4AAs. How does the judge make the final call when the macro data offers no distinction?
When the macro scoring matrices are saturated, the embryologist relies on microscopic granular nuance. They evaluate the precise thickness and uniformity of the zona pellucida, as a thinner zona is generally more conducive to hatching.
They scrutinize the cytoplasm of the trophectoderm cells, looking for subtle granularity or the presence of microscopic vacuoles.
Tiny fluid pockets within individual cells that might indicate slight metabolic stress, yeah. They assess the geometry of the cellular junctions. They are hunting for the most microscopic biological advantages to break the statistical tie.
This intense, high-resolution scientific scrutiny is fascinating. But we have to pivot to the human consequence of all this data.
We do.
Ultimately, all of these alphanumeric codes, the complex kinetic algorithms and the chromosomal readouts must be translated. They have to be delivered to a highly hopeful, vulnerable patient sitting in a clinical room wearing a paper gown, waiting to hear the fate of their genetic material.
It's heavy.
Bridging the massive chasm between cold lab data and human emotion is arguably the most complex part of the process.
It requires immense clinical empathy because patients are highly educated today. They research the Gardner system extensively. They understand that A represents the zenith of the grading scale, and consequently, they attach massive emotional and psychological weight to these letters.
So what does this all mean for the patient? It must be incredibly difficult to tell someone they are receiving a C-grade embryo when we've been conditioned since kindergarten to think C means average or failing.
It is incredibly difficult. When a patient sees a 3BC or a 4CC on their portal, the immediate assumption is that the embryo is defective. This creates a dangerous mythology around grading that clinicians have to constantly dismantle.
The pervasive myth is that an AA grade provides absolute biological certainty and a CC grade represents a futile endeavor.
And that's just a myth. The clinical team must engage in deep, clarifying conversations to reset these expectations. They must fundamentally redefine what the grade represents. The critical distinction is that morphological grading evaluates the probability of rapid implantation. It does not dictate the absolute capability of generating a healthy human life.
We need to linger on that distinction, probability versus capability.
Statistical probability simply means that a flawless 4AA uploid embryo possesses a very high statistical likelihood of establishing a pregnancy quickly, perhaps a 60 or 65% chance on the first attempt.
While a 3BC embryo, exhibiting a sparser trophectoderm and a looser inner cell mass, possesses a lower initial probability, perhaps a 35% chance.
But 35% is vastly different from zero. It still possesses the fundamental biological machinery required to execute the implantation cascade and drive fetal development.
Morphology is an organizational tool designed to sequence transfers efficiently. It is not a definitive crystal ball that prophesies the final outcome.
Exactly. A Cs-grade trophectoderm still contains enough invasive, highly adaptable cells to breach the endometrium, tap into the maternal blood supply, and generate a fully functional, life-sustaining placenta.
And the daily reality of the IVF clinic absolutely validates this, doesn't it? The data is full of statistical outliers that defy the spreadsheet.
It is the most humbling aspect of reproductive biology. We routinely transfer absolutely flawless textbook 4AA blastocysts, embryos that have cleared every genetic and kinetic hurdle we can throw at them, and they simply fail to implant.
The biological crosstalk between the embryo and the endometrial lining is incredibly complex, driven by transcriptomic signals and epigenetic factors that our microscopes cannot yet visualize. We can build the perfect ship, but we can't control the ocean it lands in.
That's a beautiful way to put it. Conversely, we witness the transfer of morphologically compromised, imperfect BC or CC embryos, embryos that barely met the criteria for freezing, and they implant tenaciously, resulting in the birth of perfectly healthy, neurologically typical, thriving children.
The lab is a masterclass in probability, but it is not the ultimate authority on vitality.
No, it's not.
I think the most crucial takeaway for any patient analyzing their embryo report is this. If a laboratory, operating under strict clinical protocols and utilizing advanced selection stacks, clears an embryo for transfer is because that specific embryo has demonstrated a distinct, viable biological pathway to a live birth.
Right. They do not transfer feudal cellular structures.
Yeah.
A C grade is not a condemnation. It is simply a different starting line for the exact same race.
Is a critical message. Every embryo chosen for transfer possesses inherent potential, recognized and validated by the embryologist acting as the judge. Well, this exploration has covered a phenomenal amount of biological ground today.
We really have.
We tracked the intense transition of the embryologist from a meticulous caregiver regulating culture media into a highly precise, data-driven judge wielding the Gardner morphology scale to decode cellular architecture. We broke down the profound clinical logic behind single embryo transfer, illustrating how the mathematics of cumulative probability protect both the gestational parent and the neonate from the catastrophic risks of multiple pregnancies.
We definitely dissected the advanced selection stack, too. From the laser-guided precision of PGTA biopsies identifying chromosomal aneuploidies to the continuous kinetic timestamps of time-lapse incubators screening out chaotic metabolic pathways.
And we navigated the incredibly volatile, real-time recalculations required on the morning of transfer. We mapped the entire selection matrix, demonstrating how visual, genetic, and kinetic data streams converge to answer the single most critical question of the laboratory. Which embryo possesses the highest potential to initiate a life today?
It's quite the journey.
As we conclude, I want to leave you with one final provocative thought to consider. We have spent this entire deep dive discussing immense technological stacks, lasers, time-lapse optics capturing images every 10 minutes, and incredibly rigid grading algorithms designed to quantify, rank, and predict human potential through alphanumeric codes. But despite all of our advanced hardware, despite our absolute best scientific efforts to isolate and control every single microscopic variable, there remains a profound, unquantifiable mystery at the heart of human biology. We cannot yet map the sheer, unyielding biological will of an embryo. Sometimes the underdog embryo, the one that didn't look perfect on paper, is the one that possesses the sheer biological will to become a life. Science, with all its power, can only guide us to the threshold of the door. Life itself still retains the ultimate authority to decide whether to walk through it.
The intrinsic vitality of the embryo remains a powerful variable that evades all of our algorithms.
Thank you for joining us on this unhurried, meticulous journey into the microscopic arena of embryonic selection. Understanding the profound why and how behind these complex laboratory decisions empowers everyone involved in this process, from the dedicated trainee calibrating their eyes to the Hoffman optics, to the patient waiting anxiously for the phone to ring. We hope this deep dive has brought deep clarity to the quiet, highly pressurized decisions executed every single day inside the incubator.
It has been a privilege to explore the deep science of this protocol with you.
In our next episode, we step up to the pass-through window for episode 13, the final lab step embryo transfer preparation. We'll break down the exact physics of loading a catheter, the intense security protocols of identity witnessing, and what happens in those final critical minutes before the embryo is returned to the uterus. Until then, take care.